Okay, you might notice that the screen's a little different this time, and that's because we're doing something a little different. So the skills for this, we're looking at protein sequence and structure homology, and we're really going to use systems biology to answer these questions. And that involves searching for primary structures on the internet. Okay, so we're going to explain what information can be inferred from protein primary structure information. We're going to justify the use of one-letter amino acid code in systems biology, also known as bioinformatics. And so that's using computers in order to answer biological questions. So we're also going to interpret a sequence alignment and differentiate sequence identity and sequence similarity. Okay, so first off, protein primary structure. So what protein primary structure is, and we've already talked about it a lot, is just the amino acids, just the residues in the structure. And so we get that information from genomes. So NCBI, there we go, NCBI. Genome information has been published through the National Center for Biotechnology Information. Here's the website. It's been so this is the United States repository for this information. There are other places that we can go as well. SwissProt PDB. So SwissProt PDB, yes, sure. This is another repository for structural information, but that looks more at 3D structural information. If we want primary structure information, we can get it through here, because it has the genomes, and once you have the genes, you can figure out what the protein is. Okay, so we're going to look for one protein in particular. It has an accession code, or sorry, a GI number, 2122-1904. And we can search for that. So this is a protein I've worked with pretty extensively. I published a paper on it. And if we go to proteins and click on protein, suddenly we have a racemase from Streptomyces silicolor A32, and this is based on primary structure information. So here's the gene locus, the accession number, the organism, how this was published, etc., etc., etc. And we scroll down, we scroll down, we scroll down, and here we see a bunch of one-letter codes. So what do you think this is? This is the primary protein structure, written in those one-letter codes that I know you already have memorized from the first part of this lecture. So methionine, isoleucine, glutamate, etc., etc., etc. So what this really tells us is just the order of the amino acids. Other than that, we don't necessarily know any other additional structural information. There are computer programs that we can use to predict secondary structure information. For example, the helices and the beta strands and any turns that would occur in this particular sequence. But it doesn't give us an idea of the 3D shape. And if we don't have an idea of the 3D shape, how can we have an idea of the function? Because these structure-function relationships in proteins are absolutely vital. So even though we have a primary structure, it doesn't tell us a lot of information unless we kind of know what we're looking for, unless we have something known that this structure is similar to. So, in order to get to that similar structure, we can do this. We can analyze the sequence running BLAST. So this will find regions of similarity between this sequence and other sequences, and maybe that will give us an idea of a function. So if we don't know this function, racemase isn't a specific function. So by running the BLAST, perhaps we can find something with a very similar sequence and we know its function. So I just keep all of my all of these the same, all of the parameters just standard, because unless you get into graduate school, you're really not going to need to change very much here. And you let this run for a few minutes. Okay, and when the BLAST is done, you get an output that looks like this. Query ID, blah, 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 blah. Okay, and what we see is MR-like, so this is a mandolate racemase superfamily-like. So what we can tell from the primary structure information is whether or not this protein is part of a superfamily. So superfamilies tend to have the same overall 3D shape and certain key elements in the primary structure that we can use to identify them. However, the same overall shape can perform different functions based on the catalytic residues in the active site. Okay. So here we see this racemase. Oh, look, it's got a crystal structure, and we see function annotated here as a decarboxylase. I'll tell you right now, that's not actually the function. So a problem with these online databases is that these are automatically annotated with a function based on the primary structure. So if the primary structure is, greater than X% identical, meaning that those, here, let me switch back. So if it's identical, one moment please. So if it's identical, that means that amino acid X at position, let's say if it's at position 200 in the protein sequence, if we look at the next protein sequence, it's going to have that exact same residue at that same position, even if it's maybe a different, like 202, but it's going to be really close to there. Similar would be like if we had an aspartic acid and a glutamic acid in the same position, or close to the same position. So these are both polar acidic residues, but they're not exactly the same. These two would be exactly the same. So that's the difference between sequence identity and sequence similarity. So keep that in mind when we're looking at this. Okay. And so we can see a whole bunch of sequences going down. Okay, moving up to the top. What I want to mention is E-value. E-value is an indication of how different the two sequences are. And when it's 0, it means it's absolutely perfect. So this is actually the sequence that we plugged in, and it comes back exactly itself. The crystal structure is a little bit different because the crystal structure has selenomethionine incorporated into it. So it changes a few amino acid residues, but really it's the same. So it's basically the same. So percent identity, 97% here. And the PDB is 2OVL, so we can look at that structure a little bit later. Okay, and we keep scrolling down, and we see something called sequence alignments. So the first one, remember, this is what we plugged in. So we can see, so this is what we put in, this is what it's comparing it to, and wherever there's a letter in between, that means it's absolutely identical. So this has perfect sequence identity, 361 for 361, 100% positive, with no gaps. If we scroll down, we see that there's a couple differences in the crystal structure, but that's to be expected. And if we compare, and as we go down, they're getting less and less identical, and we have some similarities. For example, here's an isoleucine, here's a valine. When we align these two structures, we see a plus. That means that those two are similar, they're both nonpolar, however they're not identical. And the gap means that there's not really any correlation between this proline and this alanine, this arginine and this histidine. Even though there could be, it's not very, very strong because the shapes are so different. Okay, so moral of this story, we can compare primary structures together in sequence alignments. So the best way to compare sequence alignments to get useful information is to compare a whole bunch of sequences from different superfamily members, especially ones that you know the function of. Okay, so this is an example of an alignment where we can compare the amino acid residues at a bunch of different positions. So that's what these numbers are, is it's at this position in the sequence. And so we've got all of these ID numbers and percent identities, and you can see it gets less similar as it goes down. And what these alignments can really tell you is if you have one that you know, like I know that this is a galactorate dehydratase, that's what this means, and I know that these positions are important. We can look at those positions in other ones that we don't necessarily know what they do and predict. So for example, if they have the same KXS motif, maybe that these residues are important. And so that's what these colors, these different colors indicate. So I know for a fact that 189D, 216E, and 242E, these acidic residues bind a metal that's needed for catalysis. We see, and without this, the reaction would not occur. We see those in all of them. Therefore, we can infer that all of these would bind the same metal. We know that 149, 151, 191, all of these other ones are involved in the catalysis. So because all of these residues are here and they're identical, not just similar, but identical, we can predict that all of these unknowns would have the same function based on this primary structure alignment. However, if we had different residues here, if this was, if these were missing or had something that wasn't even similar there, then we could predict that it would have a different catalytic function. Okay, so how would you confirm or test this hypothesis? How would you even do that? Well, typically what happens is that you make mutants. So if I think that this is important, if I knock it out, if I make this an alanine, alanine is nonpolar, it's not going to have a catalytic function. And then you make the protein and purify it and see if it still has that same function. And if it does have the function, you know it's not important. If you knock this out and the function stops, you know that it was important. So when I made mutants of these, I found out that all of these were in fact important. So all of these very likely have the same function. Okay. So this brings up a great point. Why would you use one-letter amino acid code for abbreviations? That's so that we can do these alignments. If we had to put 3 letters here, imagine how long that would take, number 1 to type and number 2 to align. Especially if you have a typo, the less information you put in, the better. So that is, this bioinformatics, the systems biology, really required the use of a one-letter amino acid code. So that's why we came up with one. And that's why it's kind of silly, because you have to have a different code for each amino acid, because each amino acid has its own special properties. Okay. So I think we've covered pretty much everything. One-letter amino acid code, interpret a sequence alignment, we started that. If it's the same residue in the same position, it probably has the same function. If it's different, it probably has a different function. So all of this feeds into the structure-function relationships that we see